is affected, right? Affecting the biological activity, right? So there are certain physicochemical properties that are important for assessing the biological activity. Here we will discuss according to your PCI syllabus or GPAT syllabus. There is one important component known as physicochemical properties affecting the biological activity. So in this, we'll start that aspect. Physicochemical properties in relation to biological activity. If we are very much clear about different important concepts underlined here, then it is easy for us to understand different classes of the drugs, right? That is where we will see some of the important critical aspects here. After discussing these critical aspects, we will move into individual category of the drugs, right? So there are a variety of physicochemical properties that are affecting the biological activity, right? Yes or no? Right. So coming to say, for example, say you have given a drug or a tablet orally. So orally you given a tablet, right? This tablet will initially undergo disintegration. Yes? Yes or no? What tablet contain? The tablet contain API and excipients, right? What is API? What is API? API is nothing but active pharmaceutical ingredient, or simply your drug, right? Simply your drug. So initially it will undergo the disintegration process. Later it will undergo a dissolution process, right? Whenever it undergo the dissolution, now the drug molecule is in what? Solution phase. What is the environment here? This is the lumen. This is GI lumen, right? Yes, sir. The entire thing is what? The lumen of your gastrointestinal tract. So, in order to form a solubilized form of the drug should have what the drug should have. What is the environment here in the gut? In the gut lumen, it is
Acidic environment.
aqueous environment, right? If you take the both overall the gastrointestinal lumen, the environment is what? Aqueous environment. So the point here is the drug molecule. Just a second, right? So as the environment is aqueous in nature, sorry, the drug must have which solubility? Aqueous solubility, right? Do you agree? Yes or no? If the drug is more lipid soluble, whether it will form a solution here? Yes, it doesn't form a solution. So this is the GI membrane. So the drug, whatever that is present here, in order to cross this GI membrane, the drug must have, what it must have? It should also have what? Lipophilicity. Yes? So the drug should have, in order to undergo aqueous solubility, the drug should have hydrophilicity, whereas in order to penetrate through GI membranes or any membranes, the drug should have what? Lipophilicity. If the drug is too hydrophilic, say if the drug is what? It has too hydrophilicity, can it cross the GI barrier? It cannot, right? If the drug is too lipophilic, it cannot form, what is that? A solution form of the drug. Yes or no? Yes? Yes.
Yes, sir.
That is why a drug molecule should have optimum, drug must have what? Sorry. Optimum aqueous solubility and lipid solubility. Okay. Then only it can be used as a drug, right? And after entering it into the bloodstream, here this is the GI membrane, followed by it will enter into what? The bloodstream. Again, the bloodstream environment is what? Aqueous, right? Again, this environment is what? Aqueous. From bloodstream, the drug reaches the site of what? Action, right? Sometimes the drug will need to pass through the site of action where there are many membranes, right? Here, in order to distribute, it is what? Distributing, right? In order to distribute in the bloodstream where it is what? Aqueous in nature, the drug should have what? Aqueous solubility. In order to penetrate in the organ, it has to cross the lipid membranes again. Got it? Again, it should have what? Lipid solubility. And not only that, if the drug, if it is, it needs to show its action in the CNS, say this is CNS. Already in the previous class we have discussed that there is a barrier known as blood-brain barrier. The drug again, in order to cross this blood-brain barrier, if it needs to show its action, it should have lipid solubility, right? And not only that, after showing its action, it has shown its action, and again it needs to be eliminated, right? I'll draw the kidney here just for our convenience sake. What is the major route of elimination? Kidneys, right? Again, in order to move from the out of this urinary tract, it should have aqueous solubility, right? So overall, the point here is the drug, in order to show its action, what it should have? What is that? Optimum aqueous solubility and lipid solubility. Say if we take the case here, if the drug is too hydrophilic, it cannot pass through the gastrointestinal membrane. That is why it is excreted in the feces. If the drug is too lipophilic, it is again not forming the solution form or the solubilized form of the drug. It will get eliminated through the feces. It never enters the physiological blood, or it never enters the bloodstream. So one important thing, in order to show all these, pharmacokinetics, that is absorption, distribution, metabolism, which occurs in the liver, as well as what we say, followed by, after absorption, distribution, action, it will show metabolism, and finally elimination. In order to show all this, the chemical structure of the drug molecule should be in a such a way that there is a balance between the aqueous solubility as well as the lipid solubility. If balance is lost, say if you have injected a drug, for example, if you have injected a drug which is highly hydrophilic, what happens if you inject a drug as it is highly hydrophilic? It will directly go to the kidney and it will get eliminated. There is no action. Yes. If the injected drug is too lipophilic, we also have what? Lipid tissues. Yes. And the drug will distribute into the lipid tissues and it will get stored there. And later, after a while, it will come back to the bloodstream and it may lead to toxicity. Are you able to understand the point? Yes? Yes or no? Yes, students? Please respond orally,
Yes, sir.
because I'm not looking into the messages, right? So I hope you got a clear-cut understanding that we need to design or discover new drugs which are not too hydrophilic, not too lipophilic, but drugs which are having optimum what? Hydrophilicity and lipophilicity, right? Please take a screenshot of this. So what are the various physicochemical properties of the drug molecules that are helpful for what we say, maintaining this optimum lipophilicity or hydrophilicity, or how we can improve these characters like absorption, distribution, metabolism, right? All these things we can improve, right? So we will look into the things on how we can improve this, right? And what are the different physicochemical properties that are useful for enhancing all these pharmacokinetic as well as dynamic and toxicological profiles of the drugs. Okay, are you done with the screenshot? Yes or no?
Done, sir.
Right. Right. So among the various, what you say, types of physicochemical properties, the key physicochemical property is, the first and foremost one is solubility. What is that? Solubility. So what is solubility? Solubility is nothing but simply the amount of what? Solute. Amount of solute dissolved in a given amount of solvent. It is called as what? Solubility or solubilization, right? So solubility is important for, whether it is important for absorption, important for, whether it is important for distribution, yes,
Yes.
as well
Yes, sir.
as it is also important for metabolism, or
Eliminate.
distribution, uh, metabolism, elimination, as well as to interact with the what? Target receptor. Right? All these are the different factors that depends upon what? Solubility. Right? And one important thing is the drug must have, already we have discussed, the drug must have, what it must have? Optimum hydrophilicity and lipophilicity. Right? Say if a drug has high aqueous solubility. If a drug has what? High aqueous solubility and poor lipid solubility, what will be the condition? The drug cannot cross the lipid biological membranes. All the biological membranes are rich in lipoproteins, right? Including the blood-brain barrier, right? And on the other hand, the drug has high lipophilicity and poor aqueous solubility. So when it has high lipophilicity and poor aqueous solubility, then there is no dissolution. And what is the other factor that is influenced? Distribution. Both these properties of the drug molecules will be disturbed. Please take a screenshot of that. That's why we will modify drugs in order to improve their solubility. There are cases where we will decrease the solubility. There are cases where we increase the solubility, right? We'll see different examples for that. You please take a screenshot of this. Then we will go with the different case studies by taking special drug molecules into consideration. If you are done,
Over,
please
sir.
let me. Right? Can we move further? Yes, dear students.
Yes, sir.
Okay, thank you. And we will take case 1 first. In this case, we are trying to increase the water solubility. Water, or what we say, aqueous solubility, right? So for this, first and foremost thing is, we can increase the water or aqueous solubility of the drugs through salt formation. Through what is that? Salt formation. Say for example, if you take one important drug molecule, what is this molecule? What is the name of this drug molecule? This molecule is called as, whether it has chlorine or not. It is chlorine. How many carbon atoms are there? 1, 2, 3. Chlorpromazine. It is chlorpromazine. So whether this chlorpromazine molecule is hydrophilic or lipophilic? So already I told that for lipophilic there should be what? Alkyl groups, alkenyl groups. Yes or no? In the previous class, the rings like benzene rings, all these will increase the lipophilicity. And halogens like chlorine, fluorine, bromine, iodine, they will also improve the lipophilicity. Whereas the polar groups like OH, COOH, as well as CO, as well as SH, NH, all these will improve the what? Hydrophilicity of a molecule. So if you see this molecule, how many carbon atoms are there? 6, 6, 12, 13, 14, 15, 16, 17. How many carbon atoms are there? 17 carbon atoms are there. And one chlorine atom is there. So based upon all this, what are the polar groups that are present here? One nitrogen. These are the lipophilic groups. It has two nitrogens as well as one hydrophilic group, sulfur. So if you compare this, the hydrophilicity is less or more, or lipophilicity is more. Which one is more?
Lipophilicity.
Lipophilicity is obviously more, right? That is why this molecule has poor aqueous solubility. Chlorpromazine has what? Poor aqueous solubility. That is why this molecule is treated with, whether it is base, whenever you see INE in a molecule, that means it contains some basic nitrogen. This is acid, right? And whenever it is treated with hydrochloric acid, it will result in the formation of, sorry, this is called as, there is hydrogen and chlorine, right? This is chlorpromazine, hydrochloride. This is what? Salt. Whether this is polar, what is this?
Polar.
This is what? Salt. It has positive pole and negative pole, right? Salt has what? Positive and negative poles. The salt is soluble in, what's that? Water. Is it clear? Yes or no? Yes?
Yes, sir.
Please take a screenshot of this. One approach is by forming what? The salt has good aqueous solubility, right? So which type of drug is this? This is a basic drug, right? The basic drug is treated with acid. Did you can you easily remember this structure? This is, what is this? Chlorine. Pro represents how many carbon atoms? 1, 2, 3, right? And it has how many methyl groups are there? There are two methyl groups. M represents here methyl groups, and azine represents, this is what? Thiazine, the ring, right? And this ring is called as, what is this ring called as? Phenothiazine. What is this ring? Phenothiazine. And phenothiazine ring, the compounds containing phenothiazine ring, generally they have, can I write? Did you took the screenshot? I'll write here, no problem. Just a second. You just remember, here I am trying to what? Make you understand whenever you see a ring, it is having antipsychotic activity as well as compounds containing phenothiazine also has anti-cholinergic activity. Which activity? And it also has anti-histaminic activity. So these activities are for which ring? Phenothiazine ring, not for chlorpromazine. Okay, chlorpromazine is only an which agent? It is an antipsychotic agent used to treat what? Schizophrenia. Yes or no? Right? But wherever you see phenothiazine, the compounds containing phenothiazine ring having which properties? Antipsychotic property, anticholinergic property, as well as antihistaminic property also. Clear? Yes?
Yes, sir.
Right. Please take a screenshot of this. We'll move into the other case study. By improving the solubility of acidic drugs. Right? And the next one is salt of acidic drugs. What is this drug molecule? Any idea? Yes? This molecule is called as valproic acid. Wherever you see generally val, you remember that the compound will be having 5 carbon atoms. How many carbon atoms? 5 carbon atoms. So if you see this molecule, this is, 1, 2, 3, 4. There is a methyl group. Whenever we won't write something here, that means there is a methyl group. There is a methyl group here. So how many carbon atoms are there? 5 carbon atoms. And how many carbon atoms are here? 1, 2, 3. This is, what is this? Propyl side chain. Right? This is 2-propyl pentanoic acid. Valproic acid is what? 2-propyl. The total number of carbon atoms are 5, right? That is why it is what? 2-propyl pentanoic acid. How many carbon atoms are there in total? Say this is a polar group, the carboxylic acid. The rest is what? 1, 2, 3, 4, 5, 6, 7. You have 7 carbon atoms, which is what? And 1 polar COOH group. That is why valproic acid is what? Lipophilic or hydrophilic? Yes?
Lipophilic.
Lipophilic. If you incorporate some 2 hydroxy groups here, then it will be having both hydrophilicity and lipophilicity. For this sake, we will treat it with what? A base. Are you able to assimilate the structure based on the name? Yes or no?
Yes, sir.
It is easy for you to remember just based upon the name. So now it has formed a salt, right? There is loss of water molecule, OH plus H plus, and there is formation of, this is what? Sodium valproate. It has Good aqueous solubility, right? It has good aqueous solubility. So what is the use of sodium valproate or valproic acid? It is used as an anti, I'll say it as a broad spectrum anti-epileptic agent. What is the meaning of broad spectrum? So why epilepsy? What happens in epilepsy? Can you please take a screenshot of this? Yes?
Over, sir.
Yes. I'll write the content here. Right. So what happens in epilepsy? There is
Current
rapid and
shocks.
excessive firing of neurons. What is the meaning of rapid and excessive firing of neurons? What does it mean? Say generally firing
And
of neuron means the nerve is releasing what? Neurotransmitter, right? Yes or no? Yes or no? Say this is a nerve cell. For our convenience, I have written it in the form of a circle, which is not the actual shape. So whenever there is firing of neurons, there is release of what? More amount of the neurotransmitter. So in order to release this neurotransmitter, the nerve cell also has, what is this channel? It has potassium channel, sodium channel, and we also have what? Calcium channel. Yes? Yes or no? Yes or no?
yes, sir.
Right. So whenever the sodium channel is active, the sodium channel is what? Active. It will stimulate the calcium channel, right? Yes or no? The stimulated calcium channel, there is increase in, I think I need to draw a bigger one. Say I'll write this. This is your potassium channel. This is your sodium channel. And this is your calcium channel. Whenever the sodium channel is what? Active, what happens? There is increase in the intracellular sodium levels. This will stimulate calcium channels. It will increase calcium ions. Whenever calcium ions are more, there is what? What there is? Contraction of vessel, nerve cell. Yes or no? Due to contraction of nerve cell, what will happen? There is release of neurotransmitter. Yes? This is the usual process that will occur. So in epilepsy, there is, I told that, rapid and excessive firing of neurons. When there will be rapid and excessive firing of neurons, when there is overactivity of what? Sodium and calcium channels. Yes? Yes or no? If you have more
Yes,
sodium
sir.
and calcium activity, the neurons are rapidly firing the neurotransmitter. Whenever the neurotransmitter levels is high, what happens to the balance of our physiological system? The balance of the physiological system will be lost. Whenever the balance is lost, automatically it may lead to what? Seizures. Whenever the seizures are high, ultimately leads to paralysis of the patient, right? So whatever the anti-epileptic drugs you take, generally the anti-epileptic drugs will inhibit sodium channel. There are certain anti-epileptic drugs that will inhibit what? calcium channels. Got it? Yes or no? Did you heard about GABA? What is GABA?
Yes, sir.
GABA
Gabamine.
is an inhibitory neurotransmitter. Our medicinal chemistry class will be a mixture of both pharmacology and medicinal chemistry. Okay? It is a which type of neurotransmitter? Which type of neurotransmitter?
Inhibitory.
Inhibitory. Say if there is a GABA receptor here, if this GABA receptor is active, what is that? GABA receptor is active or inactive?
Active.
Active.
Active. What will happen to the nerve cell? As it is an inhibitory neurotransmitter, it will decrease firing of what? Neurons.
Neurons.
Yes or no? It will decrease the firing of neurons. So whatever the anti-epileptic drugs you take, generally they show action either by inhibiting the sodium channels, inhibiting the calcium channels, or activating the GABA receptor activity, or promoting the GABA activity. Yes? These are the three ways how most of the anti-epileptic drugs will act and helps in the treatment of what? Epilepsy. Is it clear, the point, right? Yes or no?
Yes, sir.
You please take a screenshot of this. We will go to valproic acid. If you are done, please let me know.
Done, sir.
Right. So I told that valproic acid is an, what's that? Broad spectrum
anti-epileptic
anti-epileptic agent, because it will inhibit sodium channels as well as it will inhibit
Calcium channels.
calcium channel, and also it will activates
GABA receptor.
GABA activity, or it will promote GABA activity. As it has three types of actions, it is considered as an what? Broad spectrum anti-epileptic agent. This particular point has been asked in NIPER nearly 6 to 7 times, if I remember, right? As per my knowledge, it has been asked around 6 to 7 times. Which of the following is a broad spectrum anti-epileptic drug? Right? Not only that, what is mania? In mania also there is excess of what?
Release of neurotransmitters.
Right? Excess of neurotransmitter, right? We will call it as bipolar manic depression. So valproic acid is also used to treat what is that? Bipolar manic depression. So these are the advantages of valproic acid. It is used as an anti-epileptic agent, and it is also used as a drug for the treatment of what? Bipolar. Why we are calling it as bipolar? We are calling bipolar
Mania and depression.
mania and depression. Mania is what? Opposite to depression. In patients with this bipolar disorders, sometimes they will be overexcited. Sometimes they are much depressed, right? Due to imbalance of the neurotransmitters, right? But this valproic acid will correct the neurotransmitter levels in our physiological system and can help in the treatment of what? Bipolar. That is why bipolar means two extremes, right? We are calling dipole, polar molecule. It has positive pole and negative pole. Similarly, mania and depression. These are the two extreme poles of a behavior of a what is that? Human being, right? That is why it is called as bipolar manic depression. I hope the point is clear, right? Yes, students,
Yes, sir.
Yes, sir.
please take a screenshot of this. You're done?
Done, sir.
Right. And not only that, one is we formed a salt. You can also improve solubility through size reduction. Yes or no? This is less of chemical nature but of more physical in nature, right? So for example, griseofulvin. What is the use of griseofulvin? It is used as an antifungal agent that inhibits tubulin polymerization, right? Inhibits tubulin protein. The griseofulvin has poor water solubility. So this is subjected for micronization. Micronization is a process where we will what? Decrease the size. The name itself is suggestive, right? Micronization. There is formation of micronized griseofulvin. What happens whenever the size is reduced? Say this is the griseofulvin. Its size is reduced, right? The surface area will increase, right? Micronization will increase the surface area. Micronized griseofulvin has good aqueous solubility, right? Yes or no?
Yes, sir.
Right. What is a spiro compound? Any idea about a spiro compound? Already, I hope in chemistry class, pharmacognosy class, I have discussed about the spiro compound, right? So, griseofulvin is a type of spiro compound. If you see the structure of griseofulvin, then you can also appreciate why it is that much lipophilic. Right? You have a methoxy group here and a methyl substitution here. And a chlorine here, 2 lipophilic. Alkoxy groups will increase what? Lipid
Lipophilicity.
solubility, right? Alkyl, alkoxy, everything, right? This chlorine will increase the lipid solubility. The total number of carbon atoms is 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. Oh, it's 2 lipophilic, right? And you also have what? Chlorine. And additionally, you have methoxy groups and a methyl group. And the number of polar groups is less. This is one of the important polar group, and this is another polar group. And all these functionalities make this compound more lipophilic. I told that spiro compound is nothing but, say, so these are two rings, right? 1, 2, 3, 4. If you see both these rings are sharing a common carbon atom, right? This is called as what? Spirocarbon, and this is called as a spiro compound. If you see here, you have a cyclohexene ring and an what? Furanone ring, dihydrofuranone ring. And both these rings are sharing a common carbon atom. This is nothing but a spirocarbon. That is why griseofulvin is an example for spiro compound. Already previously we have said about, if you remember, spironolactone. We have discussed in the pharmacognosy class. Yes?
Potassium diet. Sparing.
Yeah, potassium sparing diuretic, right? So please take a screenshot of this. We can reduce the size and improve the aqueous solubility, right? If you are done, please let me know.
Done, sir.
Right. And we also have cases. The second case is, in the previous case we have increased the water solubility. Is it really required?
Yes,
Yes
sir.
or no? Say for example, there are certain drugs which are bitter in nature, right? Yes? Yes or no?
Yes, sir.
So whenever you give drugs orally, they are bitter because they are soluble in oral cavity. In such a case, to mask the bitter taste, to mask bitterness, we must, what we must? Decrease solubility. Okay? We'll see one more example for this. Right? This molecule is known as chloramphenicol. Did you heard about chloramphenicol? Yes or no?
Yes, sir.
It is an antibacterial, antibiotic.
Non-spectrum antibiotic.
It is an antibacterial antibiotic, right? Yes or no? And
Yes, sir.
antibiotic means it is natural or synthetic generally.
Synthetic, sir.
Antibiotic is generally a natural compound. Okay? It is obtained from some Streptomyces species. Okay? One of the importance or significance of chloramphenicol is there are very less compounds which contain nitro group in their structure, which are of natural nature. This is the important, or we can say, first drug natural drug with nitro group. This is one of the speciality of the chloramphenicol molecule. So the problem with the chloramphenicol molecule is, there is a small variation in the structure. There is a CH2OH. I have written the chloramphenicol is what? Having good aqueous solubility, and it also has what? Bitter taste. In order to overcome this bitter taste, this particular part of the molecule is treated with palmitic acid. So what is palmitic acid? CH2, we can write 14 carbon atoms and COOH. This is palmitic acid. Whenever acids will react with alcohol, right? And they will form esters, right? Whenever both these reacted, right? Whoa. How many carbon atoms are there? C double bond O, CH2, 14, and CH3. And there is a dichloroacetamide. This is dichloroacetamide group, and this is the nitro group. Okay. Sorry. Okay. This palmitic acid, and this molecule is chloramphenicol palmitate. It has how many carbon? 15 carbon atoms. Whether it is soluble in the oral cavity? Whether it is soluble in the oral cavity?
No, sir.
It is not soluble because of increase in the hydrocarbon chain length, right? You please take a screenshot of this. And again, whenever it reaches the body, it will undergo there are esterases. They will break this ester linkage and they will deliver the chloramphenicol back. What is the limitation of chloramphenicol whenever it is given to children or neonates? It causes a syndrome known as
Grave baby syndrome.
gray baby syndrome, right? Because it is not well metabolized in what? Neonates, right? As well as the children. Gray baby syndrome in neonates and children. Because the enzymes which are required for metabolizing the chloramphenicol molecule are not sufficiently developed in the neonates and children. That is why it is contraindicated in children. Gray baby syndrome involves, there are multiple things in the body that will change, where there is formation of deoxygenated blood. Okay, entire body will be changing the color. There are multiple problems in gray baby syndrome, right? Are you done? Yes.
Done, sir.
Right. So next important, these are some of the applications of what we say solubility. Next, coming to the other important physicochemical property, that is partition coefficient. So what is partition coefficient? Why partition coefficient is important? Partition coefficient is required for a drug to cross biological membrane, right? Yes or no? To cross,
Yes, sir.
what type of biological membranes are that? They are lipid biological membranes. So drugs partition coefficient is important as it must cross the lipid biological membranes. So partition coefficient is an indication of, as I told, it is required for crossing the lipid solubility, lipid biological membranes. It is an indication of lipophilicity of a drug molecule. Do you agree? It is
Yes,
an
sir.
indication of what? Lipophilicity of a drug molecule. Right? So generally, what is the general definition of partition coefficient? Partition coefficient is defined as the ratio of drug concentration in lipid phase to the ratio of the drug in aqueous phase. Right? Yes or no? Lipid phase,
Yes, sir.
or we will call it as
Organic.
organic phase. Right? Or aqueous phase or polar phase, we can use any other terms, right? So partition coefficient is generally given by log P is equal to CO by C aqua. CO is concentration of drug in organic phase, whereas C aqua is concentration of drug in aqueous phase. So what did you understood from this relationship? This log P is nothing but what? Partition coefficient. Here you can see that log P is directly related to CO. Yes, whereas log P is inversely related to C aqua. Yes or no? Yes or no? So what we can say? What we can say? More is the log P value, higher is the lipophilicity of a drug. Do you agree?
Yes, sir.
They are directly related, right? If the log p value is high, means it is self-understood that more drug is distributed into the organic phase. Higher is the lipophilicity of the drug, right? Please take a screenshot of this.
Over, sir.
Right. Can we say partition coefficient as an additive property? Yes, because it is additive property because why it is additive property? Because all the lipophilic groups in a drug contribute to the overall lipophilicity. Yes or no? Just now we have seen it has 15 carbon atoms, 17 carbon atoms. We are counting everything, right? Yes? All the groups are what? Contributing to the overall lipophilicity. That is why partition goes. Similarly, solubility is also a additive property. You have polar groups, all the polar groups are contributing to aqueous solubility. Similarly, our polar groups are contributing for what? Lipid solubility also, right? All lipophilic groups combine to form the overall lipophilicity. That is why partition coefficient is usually considered as what? An additive property, right? So you know what are the groups that will improve the partition coefficient property? Hydrocarbons. Yes. What are the hydrocarbons? Alkyl groups, alkenyl groups, alkynyl groups, cyclo alkyl groups. Yes or no? Cyclo alkenyl groups. Just now we have seen in brecciofulvin, which contain a cyclohexene ring. Yes? Yes or no? Similarly, what are the other groups that will improve the lipophilicity of the molecule? Halogens, right?
Halogens.
Chlorine, chlorine, bromine, iodine. Similarly, alkoxy
Dots.
groups, right? Wherever you see OR, right? As well as esters. Ester will also improve the what? COOR. Because of this R, the hydrocarbon, there is increase in lipophilicity, right? And what are the other groups, right? So these are the important groups which will contribute for what we say. All these groups will increase partition coefficient. On the other hand, OH, COOH, NH2, SH, yes, these are what? Which type of groups? Polar groups. These are non-polar groups. These groups are very much important for you to understand, because based upon this you can assess the properties of the drug molecules, right? These polar groups will, what they will do, increase the PC or decrease the PC, partition coefficient? They will decrease
Decrease.
partition coefficient. Please take a screenshot of this. We'll see some applications of partition coefficient where it has improved the biological activity.
Over, sir.
Right. So coming to the applications, some key examples. Am I writing more?
Sir?
Am I writing more or it is useful? Because what I felt is, as it is not a live, one-to-one, what we say, what is a physical class. If it was a physical class, the story will be different. But I am trying to write more so that you can take a screenshots and you can prepare from that without referring to any other textbooks also. Right? That is why I am trying to write more. No problem, right?
Sir.
Right, okay. So the first one is barbiturates to thiobarbiturates, right? This is the structure of barbiturates. The barbiturates generally they are less lipophilic. Whenever this oxygen is replaced with carbon double bond sulfur, if you see carbon double bond O and carbon double bond S, the carbon double bond O is more polar, whereas carbon double bond S is what? Less polar. Less polar means more lipophilic or not?
Yes, sir.
Less polar means what? More lipophilic.
More
This
lipophilic.
is what? Thiobarbiturates. They are having or they are more lipophilic. As there is, sorry, we need to write this double bond O, right? As they are more lipophilic, can they enter into the CNS fast?
No, sir. Slow.
Because they can readily cross the blood-brain barrier, right? So this is the blood-brain barrier. You have compound C double bond O, C double bond S, which can easily cross this blood-brain barrier.
Sulfur.
The sulfur-based compound, right? It can readily cross the blood-brain barrier. As it readily cross the blood-brain barrier, it will show the action and it will readily get out of the blood-brain barrier. That is why it will enters CNS readily. As it enters CNS readily, it has short onset. It will show the action readily, right? Short onset and short, sorry, short duration of just a second. has short duration of action. Right? Is it clear?
Yes, sir.
Just a second. So if you take some of the barbiturates, known as, we have classification of barbiturates known as short acting, long acting, intermediate acting, and ultra short acting, right? We take ultra short acting barbiturates. They acts within seconds, and their duration of action is 30 minutes, right? What are the examples for this? The examples for this is methohexitol, right? As well as thiopental, methohexitol sodium, thiopental sodium, and thiamylal sodium. If you see the structure of these drugs, the thiopental sodium, for example, if you take, or I am drawing the structure of thiopental itself. It has a ethyl group, and there is an, we are calling it as what? Pental, right? How many carbon atoms are there? 1, 2, 3, 4. There is a methyl group here, right? 1, 2, 3, 4, 5. This is pental. Here it has, what is that? Carbon double bond S. That makes this compound what? This type of acting. Yes,
Not acting.
it is what? Short acting, right? I hope you got clarity about this, right? Similarly, there are also other drugs like intermediate acting, long acting, short acting. So again, it depends upon the lipid solubility or the partition coefficient. You please take a screenshot of this. Are you done?
Over, sir.
Right. Did you heard about general anesthetics? The second application of did you heard about general anesthetics? Yes.
Yes, sir.
Can you quote any examples of general anesthetics? What is this?
Cyclopropane.
Cyclopropene.
Cyclopropane. Right. This particular cyclopropane has general anesthetic property. Why it has, whether it is polar or non-polar? It is a hydrocarbon,
Non-polar.
right? Non-polar. Whenever it penetrates into the CNS, it has what? General anesthetic property. Okay. Similarly, we have many compounds. The general anesthetics, they are not only the hydrocarbons, and most of the general anesthetics are chlorofluoro, or I can say, I'll write like this, it's better. They are halogenated hydrocarbons. Which type of hydrocarbons? Halogenated hydrocarbons. As well as you can also take ethers. Ethers are having anesthetic property, yes or no?
Yes, sir.
Right? They have anesthetic property, right? So for example, this is one example for the hydrocarbon, that is cyclopropane. This is having good partition coefficient, and it will cause what? General anesthesia. If you take this ethers, for example, you might have heard about what you say, diethyl ether, right? What is diethyl ether? It has two ethyl groups. And did you heard about chloroform? We know that chloroform is also an what? Anesthetic. There are compounds which contain both halogen as well as ethers. There are ethers we have observed. Like for example, if you take methoxyfluorine. What is that? Methoxyfluorine. The methoxyfluorine contain a methoxy group. It is ether or not? Yes? Yes,
Yes,
or
sir.
no? And it has two fluorine atoms and two chlorine atoms. This is the structure of what? Methoxyfluorine, which has a methoxy group, as well as I told that halogenated hydrocarbons. So simply you remember CN, general anesthetic is which type of property? CNS depressant or not? In anesthesia, what happens to the patient? Is the patient is active? Patient is active or inactive?
Active.
Yes, inactive. That means his CNS is depressed or activated? Depressed, right? So general anesthesia is a type of what? CNS depression. Similarly, you take other drugs like enfluorine. Did you heard about enfluorine? The enfluorine molecule is also a both halogenated and ether, right? The enfluorine molecule is very similar to this. It has a fluorine, hydrogen, just like here, and a fluorine atom. And not only that, it also has fluorines again here, a fluorine, a chlorine, and a hydrogen, right? This is the structure of what? Enfluorine. So overall, what is the thing? All these compounds are having general anesthetic property because of good. What is that? Lipophilicity, right? I told that halogens will increase the lipophilicity, ethers will increase the lipophilicity, hydrocarbons will improve the lipophilicity. As these compounds are having all these characteristics, they are generally used as what? What's that? General anesthetics, right? And similarly, you can say compounds which are having these groups are generally having CNS depressant property. If you take bromides, bromides are also having CNS depressant property, right? Okay, please take a screenshot of this.
Over, sir.
Right. And the partition coefficient, this is also a previously asked question, is determined by a nocturnal water system. What is that system? What is this nocturnal? This is the structure of nocturnal, right? One, two, three, four, five, six, seven, eight. One, two, three, four, five, six, seven, eight, right? This is what? N-octanol. So it is like your phospholipid, right? Yes or no? The biological membranes are what the biological membranes contain? What they contain?
Same.
What they contain? This is what? Phospho, and this is, which portion is this? Lipid.
Phospholipid bilayer.
Yeah, phospholipid bilayer, right? Bilayer, right? Similarly, the phosphate group here is what? Polar, right? And this hydrocarbon is polar or non-polar? Non-polar. This is polar and this is non-polar. Whether it is like a biological membrane? Yes or no?
Yes, sir.
It is not exactly the same, but it is mimicking the biological membrane. And the other one is what? Aqueous portion. So what we will do? We'll just take a separating funnel. In the separating funnel, we will add what? N-octanol and what? Water, and we'll add the drug molecule. It will be distributed here between these two phases, right? This is the lipid phase and this is the what? Aqueous phase. We'll see the concentration of drug in the lipid phase as well as concentration of drug in the water phase. If more drug is distributed into the octanol phase, then we say that the molecule is having what? Greater lipophilicity. If more drug is distributed into the aqueous phase, we'll say that it has what? What's that? Less lipophilicity, right? Got it? Yes or no? Yes. And we also have other systems and this is widely used. You can say why it is widely used. Other systems like benzene water system, as well as we also have chloroform water system, and a hexane water system. Can any of these mimic the phospholipid, or N-octanol will mimic phospholipid? Which one will mimic? Which system?
Nocturnal.
The N-octanol water system, right? Because it is more like a phospholipid. The benzene is like this, right? It doesn't have a polar group, right? The chloroform is highly non-polar. It is hang group like this. Hexane is C6H14. There is no polar group again. As they are not more mimicking like a phospholipid, this is the widely used system. They will ask a question like this: which of the following system is widely used for determination of partition coefficient? What is that system?
Nocturnal water
What is that
system.
system? N-octanol water system. Not only that, it is also determined by HPLC as well as TLC, right? They are also used to determine the what? Partition coefficient of a drug molecule. Is it clear?
Yes, sir.
Please take a screenshot of this. Shall we continue or we will stop? Students, as per your
Yes, sir.
wish. Shall we continue, or because it is almost 8, 1 minute less, it is up to you. What shall we do?